first reassuring part is to say, well, I'll be interested in the structure of cell nuclei, and in fact, of interphase cell nuclei, and that for the special case of Drosophila, which is, of course, the model organism that we've heard already a lot about during this meeting. In addition, I'll be interested in looking at these cell nuclei for different stages of embryonic development. And that has the advantage that we get to look at the same set of chromosomes under very different conditions. Now, that wasn't my idea to do that, but the whole thing came out of an A&R project that we had with Cédric Daniel and with Giacomo Cavalli for the experimental part. And so that's where the inspiration for this came from. And what I'll talk about today is the modeling part, the modeling sort of at the scale of the entire nucleus. So there's another word which is important in my title, or let's say it's important to me. It's the word understanding. Right? So a little bit like what Julien did, we can ask ourselves what we mean by understanding. And one way of understanding is, of course, with this bioinformatics way, where you would say, well, I need to look at the genome, and then I will spot things like a disease somewhere in the code. And of course, that could be extended to the epigenome. Now, I think for the people who participate in the school, we all think that the structure plays a role in this as well. And then to me, and admittedly, I'm just a physicist and not a biologist, but the message from biology is details matter. I mean, details matter quite obviously because they are the ones that are responsible for the differences between species or between cells in a multicellular organism. And of course, they matter also if we ever think about medical applications of what we care about. If we can now say, well, then we take to heart all details and we try to model these systems bottom up, that's another question. So I will make it a bit easier for me by saying, well, I will not try to understand everything. I will just try to understand the large scale structure of the interphase nuclei. And sort of the two dominant features of this large scale structure we've already encountered several times. So the first is the existence of territories. So the fact that different chromosomes do not really mix during interphase. And the second of compartments, so the segregation between U and heterochromatin. That is sort of an orthogonal mode of organization of the large scale structure. So if I want to understand now the large scale structure, what I will do is I will turn to polymer physics, and I will ask myself what polymer physics can sort of teach us about how we should go about doing this. And polymer physics teaches us that when we look at such a bowl of spaghettis, the structure of the chains in there and their dynamics, that we have to watch out for topological constraints. By that, I just mean that two chains cannot, or let's say in chemistry, or not so easily in biology, cross each other, and that has consequences. There's another thing that in these long chain systems, there are very long timescales that emerge and that we have to take into account. And there's a third aspect, which is that you can have structure formation as the consequence of very weak interaction. That's also a specific feature of what polymers can do. And there's a fourth message, which is that if one wants to understand these things, then they are universal in the sense that these are features shared by all polymeric systems. And then one does not always need to take into account all microscopic details of the system in order to describe them successfully. So another take on understanding is a quote that I like quite a bit from Richard Feynman. And he said, what I cannot create, I do not understand. That's a sort of a message that he gave to his students. Now, in my case, it's sort of a similar message, but I would say what I cannot model, I do not understand. And so what I want to do today is it's going to be two parts. One part is I will try to explain a little bit these messages from polymer physics and what they tell us about how we should go about modeling interface nuclei. And then in the second part, I will try to apply these lessons to the specific case of Drosophila. So how to model this bowl of spaghetti? Well, you can take it as a picture quite literally. So what we need is we need a chain model. For that, we need to sort of specify how long the fiber is that we want to model. We need to specify its bending rigidity. It's essentially a choice of the resolution of the system that we want to study. And now you're going to see how brutal these simplifications are. If I want to make a distinction between humans, Drosophila, and yeast, then the most important difference on that level is just to say, well, the contour length of their chromosomes differs by factors of 10. That's the main difference between them. Now, in such a coarse-grained model, you can relatively easily fix the static properties to reproduce whatever happens to be seen in a biological system. The other aspect one has to worry about is timescales. And so what we did in this plot here, this work with Angelo from many years ago, what we tried to do was to map the motion of a genomic site observed in the experiment on the motion of this kind of a polymer model. And that tells you something about time step that you're effectively using, the time scales that you're effectively exploring. And these are for ordinary polymers. In this case, these are gigantic timescales. The second aspect I mentioned was this business of topological constraints. So the fact that if these spaghettis move, they can sort of locally undergo Brownian motion, but what they cannot easily do is they cannot cross each other. And that was understood by Edwards and de Gennes in terms of the tube model and rotation. And it tells you something about what the maximal relaxation time in such a system is. And when we did the mapping for the case of cell nuclei, what we found was that the reptation time, which is the longest time, in fact, that you would have to wait to fully equilibrate the system for human chromosomes, is centuries. And for Drosophila, it's still several years. So it's something that does not really happen in on biological relevant timescales. So what you see here is you see just the timescales that might matter if we want to simulate such a system. And they span 25 orders in time, which is quite remarkable. Right? So I start at the femtosecond timescale, which is what you would do if you ran atomistic simulations. So that's a typical time step there. And the largest timescale, I take this reptation time of human chromosomes. Now, what we care about is this window here in between, sort of on the scale of between seconds, minutes, maybe days. And that has implications about how we should think about such simulation. So if you run a simulation of protein folding and you run it for 100 nanoseconds, but the protein takes milliseconds to fold, then you have a result which is not really relevant on the timescale that we care about. But if you simulate chromosome folding or unfolding or whatever, in fact, what we do is we simulate the biologically relevant timescales, but we still have to keep in mind that whatever we are going to observe depends on the initial state of the system from which we start, because we do not equilibrate. But in this case, we do not want to equilibrate. Right? We do not care about how the genome looks after a few centuries, because that's just not what is relevant. What is this? What is the essential property of the starting state? Well, I would say chromosomes are not topologically linked or concatenated. Well, that's, I think, pretty obvious as a consequence of cell division. And you can make the assumption that they are not strongly internally knotted. And that defines sort of the topological state at the beginning of interface. So the simulations that Angelo already alluded to that we ran at the time was to say, okay, so now we're just going to take the simplest structures that you can imagine that fulfill these conditions, and you could say, well, I mean, we're going to fold our bead spring chain into something like a helix, and then we just let this thing run, which is what we did. So we let this run, and we just let it run and run and run and run, and we knew from the mapping that what we were doing covered roughly like 3 days in real time. And what we then saw was we saw the emergence of territories. Right? They emerged just from this very simple recipe. So from this recipe of studying the decondensation of chromosomes during interphase. So that's nice, but what made it even nicer is that we could make a quantitative comparison of spatial distances observed in fish as a function of genomic distances. And there is a pretty good agreement, and that without an adjustable parameter between what comes out of this model and what is observed experimentally. So that to me gives me sort of license to study these systems using very simple models. And it also gives us a way to map them to these systems. It also allows us to go in the way of theory and to say, well, now that we have a much simpler model to think about, can we understand it? And the most important thing that one learns there is, well, because rotation takes so long, the motion of the chain ends is not important. And that is this analogy to ring polymer mills that Angelo already alluded to. And in the sake of time, I'll just jump over that and come to this part here, which is where, again, the lessons begin for how we should model such systems. So what we observe there's a separation of timescales. There's a very long timescale, these 500 years, that it would take for human chromosomes to fully equilibrate the system, right? That's, of course, totally hypothetical. It's an extrapolation from the second scale to over there, but it just says it happens after a very long time. We don't really care about it. What happens on shorter timescales is that the systems try to equilibrate locally, and that equilibration proceeds from smaller to larger scales. And so you can estimate that sort of after a day, maybe they're equilibrated at and beyond the megabase per scale. So what does this mean for modeling? It means that, well, I think one should model these systems top down. That is, one should have a description which is robust in reproducing the large scale structure. And to that description, one can add finer scale features. So these features are, for example, that in what I've talked about so far in my spaghetti model, I did not take into account at all that the fiber properties are position dependent. Right? I mean, there's the sequence, there's epigenetics, and all of these things are, of course, very important and affect the motion and the structure of these systems. So one very nice thing is this idea that you can locally identify along the chromatin fiber epigenetic domains. And if you do so, then of course, the next logical step for a polymer person, here you see that Danielle and Cedric took that step, is to say, well, then you should not use a polymer model like what I talked about so far. You should use a copolymer model, where the effective interactions between these beads represent or depend on their epigenetic state. And that is also what I'm going to use today. Okay, so that's general lessons. Now comes Drosophila. So here's a picture of Drosophila nuclei. So in this picture, what you see is you see interphase nuclei, and you see chromosomes during cell division. So you see the sizes are approximately comparable. And in one case, of course, you can easily distinguish individual chromosomes. In the other case, in the microscope, spontaneously, you don't see much in that sense. So now for Drosophila, well, the variant that we look at, melanogaster, if I'm not mistaken, has 4 chromosomes. Chromosome 2 and 3 look like what we always think chromosomes should look like. The X chromosome is very long and has a centromere at the end. And there's a very short 4th chromosome that is also present. And if we want to, so what we're going to do is we're going to study then polymers of these corresponding sizes. And depending on how we color them, you can either see the aspect of territory formation or the formation of compartments in pictures. So I said we need to worry about starting states, and that's not the domain where I'm the big expert, but we already heard something from a big expert on this subject. So we should think about these polymers as bottlebrush polymers that result from loop extrusion. And so what we've done is we sort of tinkered a little bit to produce structures that sort of include these ideas. So here you see sort of picture of them, how they look, right? You can see the two X chromosomes in blue and so on and so on and so on. And sort of as a bit of a sort of they have the proper size, and as a bit of a more quantitative test, what you see on the right hand side is the sequence average contact probabilities on the X chromosome as a function of genomic distance, and there's a comparison between data from the Everaers lab and what comes out of sort of our way of tinkering this. So that looks reasonable. I would not say that there's any deep insight that we have to on these systems. Then the next step is to do what Angelo already did 10 years ago. So you take these systems, you put them in a box, or in our case into a sphere, and then you start to simulate. So Pascal runs these simulations and runs these simulations and runs these simulations. And the important thing is he doesn't do this just once. He does this thousands of times. And that's the way how we build up statistics when we calculate ensemble averages population. Right? So it means that these are fairly long simulations, because not any individual simulations, but because there's so many of them. Now, I told you that, well, we want to look at different cycles and states of development. And of course, on the level of these spaghetti models, that seems like an ambitious program. So what is it that we change in order to say, now we're looking at cycle number such and such? In fact, we change 4 things. One thing is that we modify the initial arrangement of the chromosomes, so they can be either sort of in a more parallelomatic order or in isotropic order. We can vary the duration of interphase, which most certainly makes sense. Then the nuclear volume changes during development, so we try to take that into account. And then, of course, we can either switch on or switch off these epigenetic state-dependent interactions. So in the first part, I will switch them off. In the second part, I will switch them on. So what happens during decondensation, or I mean, what's the difference between the initial structures and the structures that we observe during interphase? So again, here you see sequence average contact probabilities. On the left-hand side, you see data from the Cavalli lab. On the right-hand side, you see what we see in the simulations. And what's the main feature? The main feature is that sort of on the megabase pair scale, sort of you lose intra-chromosomal contacts when they start to decondense, but you gain them on a larger scale. Right? So that's sort of qualitatively what happens. And then you see differences between late embryo or the cycle 14, which is a very prominent cycle that we'll study a lot. So that's what happens sort of if you average over everything within one arm of the chromosomes. Of course, there's much more information if you start to look at entire contact maps. So what you see here is, so you see a contact map that results from a nematic starting configuration when we let this run for a while. And on the upper right-hand side, you see drawings that are a century old or so, even older, based on microscopy studies. And so what I'll do now is I'll walk you through the features of this genome-wide contact map so that you have a little feeling for what the things are that you can really see in there. Now, the dominant feature is, of course, the most contacts are along the diagonal, meaning they are between genomic sites, which are close to each other along the chromosomes. But you can also see other features. For example, these cross diagonals, they indicate, in fact, contacts between the two arms of the same chromosome, right? And so that's something that is very natural to expect. If you just look at the old drawing, Rabel would not have been surprised by this kind of a feature. So now this feature here, so that's the correlation between chromosome 3 and chromosome 2. Again, you see these two crosses, so that's contacts between the arms of these two different chromosomes, and it tells you that these chromosomes, they are not randomly arranged, but they are arranged in a manner which is sort of parallel to each other. You can see these points here. They would say, well, the centromeres in this simulation, they stayed relatively close to each other, and these would be something on the telomeres. So now these features, in fact, if you start from an isotropic starting configuration, they pretty much vanish. Okay, so now what do we see? We see that we have in cycle 12 Rabel-like features. So if we simulate them, that comes out reasonably well. We can do the same thing in cycle 14. So you see a bit more intermingling because cycle 14 is much longer than cycle 12. But if you look at late embryo nuclei, you see that these Rabel features totally vanish, and that now we better start from an isotropic state. So these black bars, in fact, I should have said that. I mean, that's in the experiments. Experiments cannot really measure contacts in constitutive heterochromatin because there are so many repetitive sequences that they can't map the reads. So I mean, they don't see these. Okay, so that's what we can get out of the generic model. Now the question is what happens if we switch on these epigenetic state dependent interactions. So we distinguish 4 states. Red are active sites, blue, they contain lots of polycomb, green, that's constitutive heterochromatin HB1, and black is just all the rest. And in what I showed you, we did not vary. I mean, of course, we search for good parameters, but I mean, it's always the same parameter. Okay, so first thing is, again, I look at these intra-arm sequence average contact probabilities. And so now if you look at the black curve and the red curve, you just don't see any difference whatsoever. So switching on these interactions didn't change anything. And that's a good message for me because, I mean, that's a very good agreement with this philosophy of saying, well, we should pursue this kind of top-down modeling where we add small-scale features to a large-scale description. And of course, that only makes sense if after you add the small-scale features, the large scales don't change so much. So that seems to be the case. So now here you see the contact map for cycle 14, and you see that there are very strong new features. In particular, what you can see is there is sort of strong segregation of the heterochromatin part from all the rest. And these are features which you can very easily see in the simulation data. And with the help of the simulation data, you also detect the same features in the experimental data because mostly they just don't see it because they don't have the heterochromatin. So that's a very nice feature of this thing here. So let me just compare it to what we had before. So that was the generic model. So now I go back to the interaction model. So you see the appearance of these squares around the centromeres, and that is very well reproduced the model here. So it's even stronger if you look at late embryo. I mean, here these features, the segregation, they are detectable in experiment, and I think this comes out very, very well in the simulation. So again, the comparison to what we had without these interactions with the interaction. So I think that really makes a lot of sense. So what this says is, well, it is very reasonable to assume that these HB1 domains, the heterochromatin, that this segregates from the rest of the chain. So you can really see this very well. Now the question is, do we also see clustering of active domains? So you can now look at these two plots and ask yourself, are the red domains more clustered on the right-hand side? You can do the same thing for polycomb or for the black domains. and maybe it is better to look at these slices through the system, so you can very clearly see in the upper row the segregation of the heterochromatin from the rest compared to what happened in the generic model. And these are always the same starting state. They would just run with different interactions. Now, for the active domains, if you look at it, I mean, you have the impression maybe there's a little bit the same for the polycomb domains. Also, there's more heterogeneity in the black domains. But the real question is, of course, does this correspond to what we see in the high C in the contact maps? So what I'm going to do now is I'm going to zoom into these maps. This is now the full contact map, but just taking out the black parts because they make the comparison always a bit harder. And so what we're going to do is we're going to zoom into the right arm of chromosome 3. And so here it is, always a comparison experiment with the lower right triangle simulation, the upper left triangle. So that looks reasonable. Now we're going to look at one corner of this thing, which for reasons that, of course, I don't understand, my colleagues find particularly interesting. So you zoom into there. That seems to make sense. We can again zoom, and the agreement isn't bad, I would say. Right? And what you see here is mostly black chromatin, and you see the association of red, that means active sites. And we're going to do the zooming again in a different region of this arm. Again, so we're going to zoom in there and here. And in this case, you see that this is the association of fairly big polycomb blue domains in our notation, which also comes out reasonably well from this description. So in conclusion, I mean, what I've shown you is I've shown you modeling of the large scale structure of interface nuclei. And what comes out of the modeling, I think, reasonably well is territories, compartments, TET-like features in contact maps, and that reasonably well, I would say. Of course, there's more things that we can measure and have measured than I can talk about now. Now, in terms of understanding, well, I mean, I put a little tick mark behind slow dynamics and topological constraints and crumpled polymers, because, I mean, that's sort of what I like. I put a little, I mean, the copolymer model, I mean, it works very well, but I don't understand the parameters as well as for the rest, so that is something that, to me, still needs a little bit more of understanding. And of course, for you is the question, does this kind of modeling add understanding? I mean, you're the judge of this, but what I would like you to, I mean, I would like to run a little experiment at you, right? Everybody is excited about phase separation in these biological systems, and so you now look at results that come out of this kind of modeling, right? And you would say, yeah, I mean, of course there's a segregation of the green stuff from the rest. Only in this model, we know that the only ingredients were the chain connectivity and, of course, the position of the heterochromatin along the chain. The other ingredient was that the chains could not cross each other, so we had slow dynamics, topological constraints, blah, 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 territories and all that. But in this model, there are no effective interactions between the green and the non-green part. There's nothing. Right? It just looks very much like phase separation. That's something that one has to keep in mind when looking at data, and I think modeling helps you to make that distinction. It just means, well, we have to be quantitative, which we are, which is one reason why this field is so exciting. And of course, there's more. I mean, if we want to close the loop, then we have to take into account loop extrusion and to go through replication to try to go back from interface again through the cell cycle, or something that we have not done at all. And that would be very exciting. And all of these other things, I mean, one glaring omission in this modeling is, of course, the interactions with the nuclear envelope. But there's more to do, which is the reason why this is an exciting field. So let me finish with my acknowledgements. I've benefited a lot from interactions with Angelo over the years. And for this project here, it was work mostly with Cedric, and the person who has done all the simulations and produced all the plots that I've shown you now in the second part is Pascal. And so we are really indebted to him for the work and the effort he has put into that. And with that, I thank you for your attention, and I wait for your questions.
Thank you very much, Ralf. So there is a question from John. John?
Hey, Ralf, nice to see you. Great talk, very interesting.
Thanks, John.
You know, early in your talk, you know, you mentioned that you were using a stiffness parameter for a persistence length or a Kuhn length, I think, for chromatin fiber of around 3,000 nanometers. And you know, this has been a source of a lot of controversy over the years. Do you consider that the best number to use for these kinds of coarse-grained simulations? Or, you know, there's been all this work by, for example, Kazumishima on suggesting a much shorter persistence length. You know, a lot about this. I was curious about your opinion on this.
Yes. So you're totally right that, I mean, originally we took these numbers because, I mean, when we started, the 30 nanometer fiber was still totally respectable.
Well, I'm not totally sure it's disrespectable, but I was curious what you thought based on current experimental understanding would be the best polymer model.
The way I think about it now is totally different. What I do now is I look at plots like this one here, which shows you the larger scale behavior. And if you look at the, I don't know if I've lost my mouse again. Can you see my mouse or not?
Yes.
So if you look at this part here, in fact, that's sort of the equilibrated behavior. It's in fact it's almost Gaussian behavior. It's sort of chain ends, chains which are short enough not to be entangled. And so from this you get something like a mean square end-to-end distance. If you have a mean square end-to-end distance and you make a choice as to the resolution, as to the fiber length that you wish to study, you get out a corresponding Kuhn length that you have to use in order to reach this behavior. And that's, so for me, this is now a totally adjustable parameter. And the nice thing for somebody who loves entanglements is that if you make this operation, if you estimate the entanglement length from the packing model, which works very well if one does PPA and everything, it's invariant under this operation, as long as these are loosely entangled chains which are flexible on the entanglement scale. So this is just an adjustable parameter now that depends on with which resolution you want to work. But if you wanted to go down to 10 nanometer fibers, it would just mean that we have much longer fibers which are more flexible, but in the end they have to reach the same large scale mean square extension as a function of genomic distance. And that's the way we couldn't tweak this for any resolution.
Okay, I get it. So the current simulations are done with the same kind of polymer model. It's basically coarse-grained chromatin.
Yeah, and it's in this case it was we decided we wanted to simulate on the 10 kb scale because that's roughly the resolution of the high C step, and then you just go through that loop and it gives you the parameters that you want to have.
So that's the same parameter you're currently using. It doesn't really reflect any particular chromatin structure.
Exactly, and that's sort of, it's not trivial, I would say. It's sort of interesting that it comes out that way. That if you say you put in more fine scale, more distance, that it does not affect the topological properties, which are the ones that I'm always crazy about.
Yeah, sure.
But that's sort of, I find that quite cute. It's not, it's interesting.
Yeah, good. Thanks.
Okay, thanks, John. Two questions. One question from Irene Farabella. So, are you planning to take into consideration also homologous pairing in your simulations?
Yeah, and of course, you know the answer that we've already done that. Yes.
Okay. And another question from Eikut Erbas. I would assume that your chromatin polymers, it's true, have uniform semi-flexibility. How do you think a non-uniform flexibility, if it would change your the pictures?
Yes, again, there's two answers to this. So one answer is to say, I'm going to study a polymer model which has different flexibilities, and Angelo has done that, for example. And then I look at the behavior that emerges in this case. The other thing is, I would say, well, what we need is we need input data that tell us what the typical polymer statistics is in these different regions, and then tweak the flexibility in the way that I was just describing in my answer to John's question, such that the observed behavior agrees with what the experiment really does. Otherwise, what these things do is they produce effective interactions. Right? You get a chi parameter just by having polymers which have different stiffness. Then again, if you say, yeah, I want to take that into account because that gives you something like this demixing that I was showing, but of course I want to, reproduce the true high C data, that means then you choose other parameters for the remaining effective interactions that the model does not account for explicitly, and then you're back to seeing what I see here. That would be the sort of the modeling loop. Right? It's if you just say, I take a model, I change something in the model, and I see what happens, you get a result. That's easy. But to do it the other way around, to say, yeah, sure, I changed something, but many things at the end of the modeling should not change, that's a bit more complicated. Right? Because we have a target.
Okay, thanks. Maybe two last short questions. So is dynamics like loop extrusion from Todor Gitchev, is dynamics like loop extrusion mechanism taken into account? And what about Topo 2 involvement?
So the answer is an emphatic no. Right? So this is what I was trying to say on my last slide. Right? Of course there is more. So if one builds minimal models, right, then of course the fate of a minimal model is always at wrong, right? Because there's so much which is not taken into account. Then if the minimal model describes the observations, that's already interesting because it says, well, maybe whatever we've not taken into account wasn't important, right? Obviously you cannot do anything without Topo 2 and loop exclusion if you want to study the condensation of chromosomes. John has shown an extremely interesting movie of how these the two chains segregate. You can't do that without this machinery. But the question is, if this machinery is dominant for the dynamics that one sees in this more boring part that we are treating. Right? This is this very special kind of Brownian motion that emerges from this, which is so different that everything would be different, which is something that I don't know and I'm very curious about. But so yes, one should do it, but in the end, you go through the modeling loop, and then with these new parameters in the more complicated model, you have to reproduce the mean square displacements that you can see. As you see, you have to reproduce sort of the sequence average structure and all, and if is after you've done that, is your system significantly different? That's a good question.